Okay, welcome back to Beaver Science. This is the next video in the sequence regarding DNA structure, and this video is related to DNA replication. It's just a real short kind of overview, and there is a couple of good videos online. I'll try to see if I can't get a link embedded in Canvas for you to those videos. But this diagram really is going to look at basically DNA replication, and it's similar to the figures that you can find in your textbook on figure 13.11. So we'll start the same way. We'll need a single piece of paper, basically in portrait orientation, and we can start by labeling this DNA replication. So at the top, this is DNA replication, and for a reference in your textbook, this would be figure 13.11. Now, a couple things hopefully from the previous video that you picked up is DNA is a double-stranded molecule, and we talked a bit about that 5' 3' type of orientation. So we're going to show just a simple 2 lines basically to represent the outside supporting portion of the molecule. Those would represent kind of the phosphate, sugar, phosphate, sugar, phosphate, sugar. And this side of the molecule is going to be the 5' side on that side, and then the 3' side on this. And then the other side, or the other strand, the complementary strand, will be running 5' to 3' in this direction. Not going to show a ton of nitrogenous bases, complementary nitrogenous bases. We'll just look at a total of 9. And so on this strand, if you come in and go show the bonds for the nitrogen bases, a total of 9. And then over here, there'll be the complementary nitrogen bases on this side. And we'll try to line them up the best that we can here. Okay, so we'll start with this sequence or series of nitrogen bases. Let's have a T here, a C, and an A. And then how about a GAT? Easy maybe to remember. And then down here, let's do CAT. So this is one side of the DNA molecule. Now we need to find the complementary nitrogenous bases over here. So remember that thymine always combines with adenine, cytosine combines with guanine, adenine, and thymine. So we're looking at this relationship here, complementary nitrogenous bases. And so just go ahead and finish out that sequence. And there would be hydrogen bonds basically between each one of these. And so between thymine, I'm going to use, I think I'll just use little dots instead of lines. It'll be easier to follow. Between thymine and adenine, there are 2 hydrogen bonds. And between cytosine and guanine, there would be 3. And so if you just want to go ahead and kind of quickly draw in those hydrogen bonds. And just as a quick reference, remember that the unequal strain on the molecule caused by the different number of hydrogen bonds between the complementary bases causes that molecule to twist or coil into that double helix structure. Now, during DNA replication, essentially what we're going to do, and Watson and Crick, I think, knew this pretty early on when they saw that this was a double-stranded molecule. They recognized, well, hey, there's a mechanism for copying this. And that's what DNA replication is. Essentially, we're just going to copy both sides or both strands of the DNA molecule. In order to do that, we have to break these hydrogen bonds. Okay, the enzyme that breaks these hydrogen bonds is called helicase. And so there is an enzyme that will come in and break these hydrogen bonds apart, and that enzyme is helicase. So helicase will come in and it will break these hydrogen bonds. Eventually it'll break them for the entire length of the DNA molecule. And what will happen is the strands will separate. And you'll hear these terms, parent strand and daughter strand. And so the parent strand, these are the two parent strands. They're the original kind of DNA molecule. And what we're going to do is we're going to break the hydrogen bonds for the entire length of the DNA molecule, and we're going to build a complementary nucleotide sequence, a complementary strand for both sides of the parent strands, and we'll replicate or duplicate this DNA molecule, which is essentially one continuous strand of the DNA molecule, and its associated proteins in the cell is a chromosome. So this strand is going to move over here, and so we'll bring it down here, and remember that the orientation on this side is 5' to 3', and we have 1, 2, 3, 6, a total of 9 nitrogenous bases on that side. And if you look here, we have TCA to start with, and then we have our GAT, and then the last 3 sequences are CAT. Okay, why that's happening, this strand of the DNA molecule is going to move over here. Okay, and so we'll draw it up over here. Same thing, we have a total of 9 nitrogen bases, and on this side we are running from 5' to 3' in this direction. Okay, what we're going to do basically is build the complementary strands for both sides of the molecule. Let's go ahead and quickly get in the sequence of nitrogen bases for this parent strand. So that's CTA. And the last one is GTA. Alright, so if we'll jump back over here real quick, and we'll draw the complementary strand for this. Now remember that the strands are anti-parallel, and so this side is going to be 5', and this side will be 3'. And we have a total of 9 of these, and so I'm just going to try to keep these as in line as best as I can. One of the things that you'll see in DNA replication and in protein synthesis is that this is what's called the template strand. And the template strand, or the parent strand, is read from the 3' to 5' direction, but we're actually building the complementary strand from the 5' to the 3' side. So this one's running this way. We're going to use it for the template, but we're actually going to read it this direction, and we're going to build this side in 5' to 3'. Thymine is going to combine with adenine, adenine with thymine, cytosine with guanine, thymine with adenine, and so on and so forth. hopefully you're getting comfortable with understanding these complementary base sequences. Okay, and we'll just do a couple of these. These hydrogen bonds get reestablished. Remember, 2 between adenine and thymine. And I'm just going to show a few of these here, and 3 between cytosine and guanine. Okay, the enzyme that rebuilds these hydrogen bonds is called DNA polymerase. One thing that you'll notice that both on, when you see an enzyme written in biology or chemistry, if it has the suffix ASE, that means that that's a protein or an enzyme that is catalyzing or speeding up a chemical reaction. The first part of it tends to tell you what that enzyme is working on. So the 2 enzymes that are important in DNA replication are helicase and DNA polymerase. All right, let's draw this other side here and see if we can't finish this thing off. And so here we have this orientation in 5' to 3' this direction. Remember, we have a total of 9 of these complementary nitrogenous bases. And here, this is the parent strand or the template strand running 5' to 3' in this direction. So we're going to read 3' to 5'. We're going to build 5' to 3'. Adenine combines with thymine, guanine with cytosine, adenine and thymine, and so on and so forth. Hopefully you're kind of getting comfortable now with recognizing these complementary base pairs. Again, the enzyme that will reestablish these hydrogen bonds, 2 between adenine and thymine and 3 between cytosine and guanine, is that one that we talked about, which is DNA polymerase. When we're done, if you go ahead and finish out, showing the hydrogen bonds. These are the hydrogen bonds. And if we don't make any mistakes, these 2 molecules basically should be a replicate of this first DNA molecule. So we've replicated DNA. If you were to make a mistake, say you missed one, a deletion, or you substituted the wrong nitrogenous base, or the 2 sides of the molecules got off one nitrogenous base, they call those mutations. And mutations can really have one of 3 impacts. They can be detrimental, they can have no effect, or they can be beneficial. And we'll talk about those maybe at a future time. And that's it for DNA replication.